In this next video, I want to spend a good amount of time talking about the action potential, and this is probably going to end up being long. I'm going to have to break it up into different segments, so just keep that in mind. Okay. But basically, when discussing the action potential, you have to remember, make sure you have a good grasp of the basic layout of the the neuron. Okay, the basic parts of the neuron. Make sure you have a good basic understanding of membrane potential and concepts like diffusion and active transport, okay, and just kind of basic understanding of what membrane potential is, okay, because without that basic understanding of those concepts, action potential is going to seem a lot more foreign, okay, so make sure that, again, before you really dig into this topic, you understand the layout of the neuron quite well, and also membrane potential and membrane transport. All right. So on that note, I just want to get that out there. But on that note, let's start discussing this topic. Okay. An action potential is basically what we often refer to as a nervous impulse. Okay. An electrical signal. Okay. What an action potential is, it's a current that is sent down the length of the axon towards the synaptic end of the axon, where these synaptic terminals or boutons are located. Okay, so bear in mind the whole point behind this is sending an electrical impulse down the length of the axon for the purpose of secreting neurotransmitters. Okay, that's how neurons communicate, via the chemical synapse. Okay, that's probably the main form of communication that neurons use. There's other forms like electrical synapses and things like that, but I'm not going to get into that. Okay, and one thing to bear in mind is that the action potential travels away from the cell body down the length of the axon. It does not go in reverse. It does not go in the opposite direction. Okay, and then again, that has to do with the layout of the neuron. All right, with where the receptors are located on the cell body, and the axon hillock, where there's the highest concentration of sodium, sodium channels in the neuron itself. Okay. Now, bear in mind, when talking about the action potential, when we're talking about generating an electrical impulse down the length of the axon, okay, you have to remember that this is caused by the movement of ions across the cell membrane. Okay, but before an action potential occurs, there has to be a resting membrane potential already established, meaning there has to be a certain concentration of ions on both sides of the cell membrane. Okay, because if we were at equal amounts of sodium and potassium on both sides, there'd be no, driving or motive force to create an electrical current. All right, and remember that resting membrane potential. When you want to understand this, remember that resting membrane potential revolves around what is the cell membrane permeable to? Which ion is this really permeable to? So think of this as like a close-up of an area of an axon. Okay. And if we take a look at this axon blown up, you're going to notice that there are, a few major types of channels, located within, on the surface of the axon. Okay, you've got sodium channels or sodium gates. You've got potassium channels or potassium gates, and you've got what's called the Sodium-Potassium Pump. Okay, or the sodium-potassium ATPase. Okay, remember it's called that because this is a form of active transport. Remember, when we talk about something like passive versus active transport, remember passive movement or passive transport is when you make a cell membrane permeable to, let's just say, for the sake of this discussion, an ion. Okay, and when there's a difference in the concentration of that ion across the cell membrane, it's going to move as long as the membrane is permeable. Okay. Now remember, there's far more sodium on the outside of the cell versus the inside of the cell. So if I open this sodium channel, if I open that sodium gate, then naturally, so let's say there's 150 sodium ions on the outside, and let's say there are 7 sodium ions on the inside. Okay. If I open that sodium gate and I just leave it open indefinitely, which it doesn't happen this way, but we're just doing this for the sake of discussion. Remember, sodium is passively going to travel down its concentration gradient into the cell. Okay, and if I leave that sodium gate open, okay, sodium will travel in indefinitely until there's an equilibrium reached in the concentration. All right, but this doesn't happen. Okay, these sodium channels do not stay open for very long. Okay, and at rest, these sodium gates are closed. Okay, so being that these sodium gates are closed, all right, the cell membrane is not permeable to sodium. Remember, when you learn about, the phospholipid bilayer, the cell membrane, remember that it's not very permeable to polar molecules or polar ions. Okay, when I say polar, think charge. Okay, so as a result, that's why ions, sodium, potassium, chloride, glucose, amino acids. Okay, that's why all those charged molecules, charged ions, they need some kind of facilitator to allow diffusion to take place, because they just can't naturally move through the cell membrane on their own because of the the basic chemistry behind that. Okay, so and remember, these gates, these channels are highly specific. Okay, remember, all these channels they're proteins that are all coiled together. Okay, and basically when we open a gate, the proteins uncoil and allow for the movement of the specific ion through there, and then when they close, they kind of coil back up. Okay, so in saying that, when the cell is at rest, sodium cannot pass in and out of the membrane. So as a result, sodium really does not influence, or sodium doesn't really directly influence membrane potential because it can't leak in or out on its own. Okay. Potassium, on the other hand, though, there are gates. There are specific gates for potassium as well. And when the neuron is at rest, these potassium gates, these proteins, are not always fully coiled or fully closed. Okay. So as a result, since there's a very high concentration of potassium on the inside of a cell, again, I'm just throwing numbers out there, 145 versus, let's say, 13. Okay, again, by your basic laws of diffusion, potassium is going to want to leak outward. Okay, potassium is going to leak out. All right, now, what's going to go on here is, remember, every time a potassium ion leaks out of that cell, the inside is going to become predominantly more negative. Okay, so by the laws of diffusion, there's a pushing force forcing potassium out of the cell. Okay, but because of all these negatively charged anions inside the cell, they're basically, negatively charged proteins or functional groups that were disassociated from these proteins that ended up becoming negative ions. Okay, the inside of the cell is very, very negative. Okay, in comparison to the outside. All right, so what's going to happen, now you know what happens when you're talking about opposing charges, okay, is that there's a pulling force there. They attract to one another. All right, so basically what's going to happen is potassium is going to slowly leak out because of this concentration gradient difference. Okay, but eventually the inside of the cell is going to get more and more and more negatively charged. Okay, and the more negatively charged the inside of the cell gets, that's going to start to exert a pulling force on the potassium. Okay, that's going to exert a pulling force on that, and eventually, at some point in time, you're going to reach an equilibrium between the pushing force of diffusion and the pulling force of the opposing charges. And then, when you reach that equilibrium and you measure the concentrations of potassium, across the cell membrane, that's when you say you've established a resting membrane potential. Okay. And typically, according to most anatomy and physiology textbooks, you're going to say that the resting membrane potential of a neuron is around negative 70 millivolts. Okay? It is around negative 70 millivolts. All right, because, remember, the inside of the cell is a lot more negative than the outside is very negative in comparison to the outside of the cell, the extracellular environment. Okay, so if I, run this electrode in there, I have a ground wire here, and then I measure the electrical charge across that cell membrane, you will get on average a resting membrane potential of negative 7 millivolts. Okay, so now when we're talking about initiating an action potential, what we're doing is we're changing this electrical charge. Okay, we're changing this by moving ions into and out of the cell. Okay, one thing you have to keep in mind about neurons is they have three very basic properties. Neurons are irritable cells. When I say irritable, think excitable. Okay, meaning they're sensitive. When I say they're excitable, they're irritable. kind of think in your head that neurons are very sensitive to stimuli. They're very sensitive to changes in their environments, okay? And it doesn't necessarily have to be a change in temperature, a change in your body fluid chemistry. Oftentimes action potentials are initiated or inhibited by neurotransmitters, by neurons communicating with one another. Okay, and I'll talk more about the synapse in a little while, but there are neurotransmitters that could make this happen. Okay, but our neurons are irritable, excitable tissues. Okay, and then once you irritate the neuron enough, it has the property of conductance. Okay, when I say conductance, what I'm saying is it's going to transfer that stimulus, not stimulus, but the excitability down the length of its axon. Okay, skeletal muscles have this property as well. Okay, or you think of, I shouldn't say muscles, okay, because when you excite a muscle, let's go with skeletal muscle. When you excite a muscle, you're gonna conduct that action potential down the length of the muscle, so you can make the entire cell function or do its job. Okay, for a muscle it'd be contraction. For a neuron, it's getting that electrical impulse down to these synaptic terminals to release neurotransmitters. Okay. And then that's the secretion aspect of a neuron. Okay, once we send that electrical message down the length of the axon, then neurotransmitters will be released. And like any other kind of chemical message in the body, I'm saying this basically, but chemical messages realistically do one of two things. They either increase activity of a cell, of a target cell or target tissue, or they decrease activity. Realistically, that's what they do. Okay. So again, I know it's kind of a basic review, but keep that stuff in mind when you're thinking about the action potential. You have to remember, like I said, understand resting membrane potential. Now, when we talk about initiating an action potential, okay, there's various different, situations in when we'll send an action potential. Now, like I said, this is the main form of communication that neurons use, okay? We irritate a neuron to the point where it generates an electrical impulse, an action potential, and it releases neurotransmitters, okay? And, basically, like I said, stimuli are going to cause this. Like when you learn about sensory physiology, when you learn about, the special senses of the body, such as things like touch, taste, smell, temperature, pressure, chemistry, pain, things like that, okay? There are various stimuli that will irritate a sensory receptor and make it generate an action potential. When you put your hand on a tabletop and you feel it's cold, okay, you're irritating thermal receptors because there's a big enough difference between the temperature of this tabletop and your hand to irritate those receptors, and they're going to generate action potentials to your brain. Your brain is going to receive this information, via specific neural pathways, and it's going to say, oh, this tabletop's cold. Okay, so this is, again, the main form of neural communication here. And again, other neurons can either, initiate action potentials or make it harder. They may, excite or inhibit the activity of, the action potential. Excitatory presynaptic or inhibitory presynaptic excitation may go on. Okay, or EPSP potentials. So